Bismillah ar-Rahman ar-Rahim. So we are discussing the antibiotic drugs and this is the second videos of the antibiotic drugs, and this is the part B of this beta-lactam antibiotic. Fine. So if you remember in the previous videos, in the part A of this beta-lactam antibiotic, we talked the mode of action, and we also see the structure of the beta-lactam antibiotic. Fine. Now this is the part B of the second videos of this beta-lactam antibiotic, and in this videos I am going to talk about the resistance which are activated against this antibiotic, and the antibacterial spectrum, and also the side effect of these drugs. Right? Now if we see the antibacterial spectrum, or we see the broad spectrum of this antibiotic, so it is activated against gram-positive as well as against gram-negative. So let me write the antibacterial spectrum here. Antibacterial spectrum, or we can say broad spectrum. So it will be activated against streptococci, pneumococci, bacillus anthracis, so bacillus anthracis, Staph aureus, Staph epidermis, E. coli, Neisseria gonorrhea, meningococci, so against these bacteria, these beta-lactam antibiotic will show good results. Fine. Or we can say these kinds of, there are many other bacteria, but these are the common. These bacteria will sensitive to this antibiotic. Fine. If we see, the gram-positive will be a large number. So streptococci, pneumococci, bacillus anthracis, Staph aureus, Staph epidermis, these are the gram-positive, and the rest of these three are the gram-negative. So if we see the antibacterial spectrum, so it will be activated better against the gram-positive as compared to gram-negative, although they will show better result in case of gram-negative as well. But here the reason behind that, in case of gram-positive, I told you that they have prepared the peptidoglycan in a large amount. So that's why it will be affected. We can say that it will go good result against these bacteria. But in case of gram-negative, these peptidoglycan will be synthesized in a low amount. So that's why, although it is activated against, it will show good result, but not best result as compared to the gram-positive. Fine. So that's all about the antibacterial spectrum. Now what are the resistance which are activated against these drugs? The resistance which are activated against these drugs, there are many ways of the resistance. Whenever I was reading these topics, and whenever I was preparing this topic, so I saw many resistance, but I will explain five. I will explain five resistance. Fine. So the first resistance which are activated against this antibiotic, this is absence of peptidoglycan. Peptidoglycan, or we can say no cell wall. What does it mean? It means that some bacteria don't have cell wall, like mycoplasma. If we see the mycoplasma, it is very smallest bacteria, and in this bacteria they don't have the cell wall. Fine. So let me write the example here. Mycoplasma. So mycoplasma will show resistance. Why? Because these bacteria don't have cell wall. And you better know, if you remember the mechanism of action of this beta-lactam antibiotic. So this beta-lactam antibiotic will interfere with the cell wall synthesis. So this is the first. The second is that blockage of Purin channel. Now what is Purin channel? Purin channel is actually a way through which the antibiotic will get enter. And these Purin channel are usually present in gram-negative bacteria. Fine. So if we see the structure of the gram-negative bacteria, so let's suppose if it is gram-negative bacteria, let's suppose this green one represent the cell, this green one represent the inner membrane, this red one is the peptidoglycan layer, and this blue one is what? This blue one is the outer membrane. Fine. And after that there are some pumps. No, sorry, not pumps. There are some channels or holes. Now, due to these channels, we call that Purin channels. Purin channels. Now, what are the function of these Purin channels? Due to these Purin channels, the drugs will easily get enter. So these are, let's suppose, the drugs. So the drugs will easily get enter. But what actually the resistance, whenever the resistance are activated, now in case of resistance, what actually happen? These Purin channels will be blocked. So these Purin channel will be blocked. So once Purin channel are blocked by these gram-negative bacteria, so the drugs will be not get internalized. Or we can say the cell membrane will be impermeable. What does it mean? It means that the drugs, they will not receive the drugs, although they will receive the essential substance, but they will not receive the drugs. Due to this way, this bacteria will be survive, and this bacteria will multiplying and dividing. This is the second one. The third one, resistance, if we talk to beta-lactamase enzyme. Now what is beta-lactamase enzyme? Beta-lactamase enzyme is that enzyme which are activated against these beta-lactam antibiotic. Here I will discuss the beta-lactamase in a detail. So what happened actually? Let's suppose if this is the structure of beta-lactam antibiotic, fine, means cephalosporin, penicillin, carbaphenems, monobactum. Let's suppose these are the structure. What happened? They will easily bind to the penicillin-binding protein or transpeptidase and diallocarboxypeptidase. They will easily bind to that area. It can easily fit. But actually what the bacteria will do? The bacteria will release beta-lactamase enzyme. And what actually happened? The beta-lactamase enzyme, if I draw the beta-lactamase enzyme here, so the beta-lactamase will be look like in this form. If we see the structure of this penicillin-binding protein and this beta-lactamase, so let me write beta-lactamase. And this is penicillin-binding protein. And this is beta-lactam antibiotic. Fine. Now if we see the structure of this beta-lactamase and this penicillin-binding protein, both are the same. Both have the same form. But actually this beta-lactamase enzyme is a little bit larger, a little bit broad as compared to this penicillin-binding protein. Due to this broadness of this beta-lactamase enzyme, they have high potency and they have high electricity. They have high electrostatic force. Due to this way, what happened? They will directly bind to this penicillin-binding protein. To this beta-lactam antibiotic. And then what happened? Let me write here. So this beta-lactamase will bind. So whenever it bind with this beta-lactamases, so due to high electrostatic force, or we can say due to high potency, they will start degrading and they will cleave the bond. They will cleave the bond. So they will start degrading this in this beta-lactam antibiotic, and this beta-lactam antibiotic will be look like in this form. So this area will be broken down from this site. So this is 6-aminopenicillinic acid site, and this is the means here the oxygen will be added. So what happens? That area will be break down. Once that area are break down, so what happens? This antibiotic will start degrading, and finally the antibiotic will be finished. And due to this way, this antibiotic will lose their affinity, or we can say they will lose their binding ability. So after that, they will not bind to this penicillin binding protein. They will fail. So they will fail. This antibiotic will fail because it is degraded in form. It is hydrolyzed. So that's why this antibiotic will be not bind. Fine. So this is the third one resistance which are activated against these drugs. If we see the beta-lactamases enzyme, this beta-lactamases enzyme, in case of gram-positive, it is produced in a inducible form, in a large amount. Why? Because these bacteria don't have the outer membrane. Fine. So that's why they are produced in a large number, as well as these beta-lactamases will also produce outside the cell. So whenever these beta-lactamases are produced in case of gram-positive outside the cell, so they will cleave or they will degrade the antibiotic outside the cell. Fine. So the antibiotic will not get internalized. Before this antibiotic will get internalized, this antibiotic will already be chopped, or this antibiotic already be cleaved by this beta-lactamase enzyme. But if we see in case of gram-negative bacteria, all of you better know that in this gram-negative bacteria, so it is gram-negative bacteria, fine. In this gram-negative bacteria, they have periplasmic space. And what are the importance of this periplasmic space? They store the beta-lactamase enzyme at that area. Once they store the beta-lactamase enzyme at that area, then they will have rapid activity against these beta-lactamase enzyme. These beta-lactam antibiotic. They have rapid activity against the beta-lactam antibiotic. Fine, because they already stored this beta-lactamase enzyme in the periplasmic space. And they will not release this beta-lactamase enzyme to outside, because they don't have because they have already this outer membrane area. And so they are not leaving, or they are not release out this antibiotic outside the cell. Right? So this is another way of this resistance of this beta-lactamases. Now there is another important thing that I want to tell you about the beta-lactamase enzyme. That in case of gram-negative, these beta-lactamase enzyme will be produced in a constitutive form, in a low number. Why? Because they already stored it into the periplasmic space. Fine? Fine. So once the resistance will activate, so this enzyme will be free, this penicillin-binding protein, or we can say transpeptidase or DLA-carboxypeptidase will free, and they will again store their function. Fine. So this is the third one resistance which are activated. Now the fourth one resistance, if we talk the fourth one resistance which are activated against these drugs, these are the enzyme alteration. Alteration. What does it mean? It means the enzyme which are used in cell wall synthesis, this enzyme will be a little bit altered or a little bit changed. If we see the structure of this enzyme, so the enzyme will be look like in this form, let's suppose. But what happened, due to some spontaneous mutation, this enzyme will be changed. This enzyme will be changed. So what happened? There will be a little bit slight changes occur at this. This is the transpeptidase site. So the transpeptidase modification will occur. So let me write transpeptidase modification. Now whenever the transpeptidase are modified, so what happened? Due to this modification, this beta-lactam antibiotic will not bind, will not fit here. they will lose their affinity, they will lose their binding ability. Due to these changes, the antibiotic will not fit here. So the antibiotic, if I draw here, this antibiotic will fail to bind here. Fine. So this is another resistance which are activated against these drugs. Now the last resistance, if we talk to, these are the production of efflux pumps. Efflux pumps. Now what are the efflux pumps? Efflux pumps are those pumps which can pumping out the antibiotic, or which can release out the antibiotic. Let's suppose if this is the bacteria and these are the Purin channel, then let me draw there with the green markers. So these green markers represent that there are the efflux pumps. These are the efflux pumps. When the antibiotic will get entered through this Purin channel, then the efflux pump have some force of attraction. Due to this force of attraction, these antibiotic, whenever they get internalized, so they will pumped out. These efflux pump will attract these antibiotic with a high force. Once they receive this antibiotic with a high force, so this antibiotic will get out. Once the antibiotic will get out from that from the cell, so how this antibiotic will perform their activity? Antibiotic will not perform their activity, and then after that the bacteria will again dividing and multiplying and survive. Right? So due to this efflux pumps, they will pump out the antibiotic. Pump out β-Lactam antibiotic. So this is the last one. Resistance which are activated against these beta-lactam antibiotic. Fine. So that's all about the resistance which are activated against these beta-lactam antibiotic. Fine. Now if we see the summary, or if we see the conclusion of this beta-lactam of this resistance activity which are activated against these beta-lactam antibiotic, so you should keep one thing in your mind, that these resistance are not activated every time. The resistance may be activated when you give antibiotic at a long period of time. When you give antibiotic to a long period of time to a bacteria, then the resistance will be activated. Otherwise, the resistance will not activate it. Keep it in your mind. Resistance will be activated when you giving the same antibiotic for a longer period of time. If let's suppose if we are the bacteria and if we are tolerating the antibiotic for a long period of time, so definitely we will get some resistance, we will get some energy from our surrounding, due to which we can compete with this antibiotic. Fine, this is the first conclusion. This is the first main important thing. The second thing, and the second main important thing, is that you should have to keep one thing in your mind: the resistance not automatically will activate it in the bacteria. They will get resistance from the surrounding or from the surrounding bacteria. So let's suppose if there are some bacteria, let me draw here. So let's suppose this blue one are the resistance bacteria and this red one are the susceptible bacteria to this antibiotic. Then what happened? This blue bacteria, let's suppose this resistance bacteria will have some genes. Among these genes, they carry this. Let me draw this green one. Let's suppose this green one are the resistance property or the resistance genes. So due to some circumstances or due to some physical changes or chemical changes in the environment, this bacteria, let's suppose, die. Fine. So this bacteria, let's suppose, die. So this resistance genes will be free, and each of these bacteria will get this resistance property. Let's suppose these one bacteria will get these resistance genes. So once these, although they are the sensitive bacteria to these beta-lactam antibiotic, but once they get this resistance genes, so these sensitive bacteria will be converted into a resistant bacteria because they gain the resistance. So here one things are clear, that the bacteria will not actually activate the resistance, they will gain the resistance from their surrounding. From this way they will gain the resistance, or they will gain the resistance if this bacteria are not die. Let's suppose, let me draw again this bacteria. If this bacteria are not die, fine, so these bacteria will come closer to this bacteria, and after that a conjugation process will occur. A conjugation process will occur, a conjugation tube will form between each of the bacteria, due to which they will carry their resistance genes to that bacteria, and then this bacteria will also resistance. This is another way. Fine. If we talk the flux pumps will not present always. The flux pumps may be created when the antibiotic will get internalized. The flux pumps, at that time the flux pumps will create it. A flux pumps will not present always. Keep it in your mind. This is another important thing. Fine. So that's all about the beta-lactamases enzyme. So that's all about the beta-lactam antibiotic and beta-lactamases enzyme and the resistors which are activated and the antibacterial spectrum of these drugs. Now, if we giving this antibiotic for a long period of time, although the resistance occur, then there are some side effects of this antibiotic also. Fine. So which one? Let me explain. These side effects are the neurotoxicity, nephritis, diarrhea, hypersensitivity. Diarrhea, hypersensitivity, nephritis, neurotoxicity. So these are the side effects of these antibiotics. There are some other side effects, but these are the common one. Fine. Then another question which also arises, these beta-lactamases, this third one resistance which I explained in detail, this third one resistance which always common, or this third one resistance will common in every bacteria. Then the question will arise in our mind that can we inhibit this beta-lactamase enzyme? If they are common, yes, the answer is yes. How? We can inhibit it when we giving the beta-lactamase inhibitor, and these beta-lactamase inhibitors are the tazobactam, clamminamic acid, or sulbactam. So let me write here. Let me erase this one area. So let me write here. So these are the beta-lactamases. Beta-lactamases inhibitor. And these beta-lactamases inhibitor are actually which one? Tazobactam, sulbactam, clamminamic acid, maybe the spelling is right or wrong, then you will check the spelling. Fine. So these are the beta-lactamases inhibitor. What actually happened? The cephalosporin, carbaphenem, monobactum, and penicillin, all these beta-lactam antibiotic don't have these beta-lactamase inhibitor. But when we add these beta-lactamase inhibitor with these drugs, any one of them, if we add the tazobactam, if we add the sulbactam, if anyone we add these beta-lactamase inhibitor, then what are the mechanism of action? The mechanism of action will be totally changed. What happened? So let's suppose this is the antibiotic, beta-lactam antibiotic, let's suppose. Fine. And they will carry what? They will carry the clamminamic acid. So let me draw the structure of the clamminamic acid here. So this is the structure of the clamminamic acid. They will attach the clamminamic acid to this beta-lactam antibiotic. What happened? Whenever they are binding to this penicillin-binding protein, so before they are binding to this penicillin-binding protein, first they will release this beta-lactamase inhibitor. So once they release this beta-lactamase inhibitor, then what happened? This beta-lactamase inhibitor will start fighting with this beta-lactamase enzyme. So let me draw with a green marker this beta-lactamase enzyme. This is the beta-lactamase enzyme. So this inhibitor will bind to it. What are the reason behind that? The reason behind that, both have the strong potency and both have the strong electrostatic force. There will be a competition start. There will be fighting start between this beta-lactamase inhibitor and this beta-lactamase enzyme. So once the fighting are start, so this beta-lactam antibiotic are free and they will get advantages from this fighting. They will easily bind here because this enzyme are fighting with this. So this enzyme will not binding here because they are fighting with this beta-lactamase inhibitor. So what happened? This beta-lactam antibiotic will bind to it. So let's suppose if this enzyme win the match, or if we see this enzyme will finish this beta-lactamase inhibitor, although it is not possible, but if let's suppose if it win the competition, so whenever they are trying their best to bind with this beta-lactam antibiotic, they are fail. Why? Because this beta-lactam antibiotic already bind. So once they are bound, so how they will cleave it, how they will degrade it. So it is not possible for this beta-lactamase enzyme. Fine. But in some cases, these beta-lactamase enzymes are also finished by giving these tazobactam, sulbactam, and clemminamic acid with the beta-lactam antibiotic. Fine. So these are another important discussion. So these are very high dose, very high dose. We usually not give this dose because there are always a lot of side effects whenever we give these combination drugs, clemminamic acid plus penicillin or clemminamic acid plus cephalosporin. It is very high dose, right? We give this dose or we give this antibiotic in a serious case. Fine. So that's all about the beta-lactam antibiotic, the resistance. Now I explained this topic in detail for this purpose because I make this topic specially for the MPhil levels and for the bachelor level student. I already explained it in a molecular level. It is not for the metric and for the FSC level student. Fine. So it is especially for the MPhil level student or for the bachelor level student. Fine. So if still you have any kind of question in your mind, so write it into a comment. I will try my best to give you answer. So if you like the video, please hit the like button, share this video, and also subscribe the channel to get upcoming videos lecture regarding with the antibiotic. Fine. So inshallah in the next video we are going to talk other antibiotic drug. Because why? Because these beta-lactamase enzyme will show resistance against these beta-lactamase inhibitor as well. These tazobactam, sulbactam, clemminamic acid are also resistant, are also not working sometime. Then there are some other antibiotic we use. Fine. We'll be talking inshallah in the next videos lecture. Fine. So please like the video, share the video, and also subscribe the channel. Thank you so much.